Hi Bio 101, this is Dr. Lennon, your favorite friendly neighborhood biology instructor, with a little pre-lab lecture about the eukaryotic cell cycle and mitosis. This should be a review for most of you. This is something you probably have covered in both middle school and high school, and I truly can remember my middle school teacher drawing this on the board in the 7th grade. Her name was Mrs. McKelvey. She had all sorts of interesting quirks. So think back to middle school, think back to what you learned in high school, and we'll just give you a little review on mitosis so you'll be ready for lab this week. Now this pre-lab lecture is in 2 parts, so you can take a little break in between if you would like to, and you can watch these as many times as you would like. So mitosis results in genetically identical daughter cells. Some of the fun things you'll notice when we get into cell division and we later get into DNA replication is there are some very feminine terms here. We have daughter cells, we have daughter nuclei. It's interesting. So if you're a sociology major or a psychology major, you might want to look up, hey, why is this such a feminine term? But if not, that's fine. The key thing with mitotic cell division is that it results in 2 cells with identical genetic information in the nucleus. These 2 cells are our daughter cells. If you look at the cells on the left, these are animal cells, and these are plant cells on the right. They're identical. We're talking about the DNA within the nucleus though, so there's a little bit difference when we start talking about mitochondria and chloroplast DNA. And we'll see when we talk about meiosis, there are also some differences when we talk about meiotic cell division. So with mitotic cell division, you get identical nuclear DNA. On the right, we see it's probably a whitefish blastula. It's an animal cell. And you can see the characteristic chromosomes within the nucleus. This is often how we picture them. The chromosomes are duplicated with cell division. We're going to talk about how that happens when we get into DNA replication. As we've said before, DNA is packaged into chromosomes, and those chromosomes are made up of chromatin. The chromatin, as we've said before, is the DNA wrapped around proteins, and those are the histone proteins, if you remember that, when we talked about chromatin in chapter 4. So when we pack our DNA, we wrap it around histone proteins, and we pack it very, very tightly. Those are the chromosomes that we think about when we see them in cell division. And the chromosomes, a good thing to remember is they're packed so tightly that you really can't even use the DNA. They're just for moving things around in cell division. All the chromosomes in a cell constitute the cell's genome. So when you hear about genome projects in the news, that's what they're talking about. They're talking about all of the chromosomes in the nucleus. A genome can consist of a single DNA molecule, so like prokaryotic cells with their single circular chromosome, or several DNA molecules. This is more common in eukaryotic cells. So as you probably know, human somatic cells, normal cells, not reproductive cells, have 46 chromosomes, 23 from mom and 23 from dad, and that's the genome. We're also going to be talking about meiosis after we do mitosis, and meiosis results in the production of gametes. Gametes are reproductive cells like sperm or eggs. They only have 1 set of chromosomes. So if you look at a human egg cell or a human sperm cell, you'd see 23 chromosomes. Once they're joined together through reproduction, that embryonic cell would have the 46. Again, that's just for humans. A somatic cell is a non-reproductive cell. They have 2 sets of chromosomes, 1 from mom, 1 from dad, 1 from each parent. The graphic you see on the right just shows you some interesting information about the size of different organisms' genomes. And if you look closely, you'll see mammals up here at the top. Here are arachnids. That's a good one for October for Halloween. Arachnids are spiders. There's fungi, the average fungus genome size. There's a protozoa, or protist. So you can see there's a lot of variation in the size of genomes. In order for eukaryotic cell division to take place, we have to make copies of our chromosomes. So in the picture on the top here, you see a single chromosome. Once the DNA is copied, this is the way we usually envision our chromosomes. So we have two copies of the chromosome, one on the left and one on the right. So each replicated chromosome is made up of two sister chromatids. So we have a chromatid here on the left and a chromatid here on the right. Those sister chromatids are separated during cell division. If you look at the sisters, you'll see a little pinched area somewhere close to in the middle. It's kind of like a waist. And that's called the centromere, and that's where their sister chromatids are most closely attached. So they're stuck together right here at the centromere. When the sisters are stuck together, we consider this one chromosome. Once we separate them in cell division, it will become two chromosomes. So here's what the sisters really look like together with an electron microscope. You have your chromosome. On each side of our centromere is called an arm. So there are two arms on the single chromosome. And remember, this is our DNA wrapped around our proteins. Once it's duplicated through DNA replication or synthesis, these are the two sister chromatids together. So we've got identical copies of our DNA. We're going to separate the sisters through mitosis so that we'll have two identical chromosomes. So the eukaryotic cell division is for production of somatic cells and has two stages. Mitosis is division of the nucleus. That's when we're going to divide the nucleus and we'll pull apart the sister chromatids and we'll end up with two identical sister nuclei. The second stage is cytokinesis, and that's division of the cytoplasm. So cytokinesis is a part that's separate from mitosis. Gametes, as we'll learn, are produced by a variation of cell division called meiosis or meiosis. I say meiosis, you can say meiosis. I don't really care. We'll get there. In 1882, a German anatomist, Walter Fleming, actually developed the dyes so we can see the chromosomes in cells. Up until that point, we couldn't actually see chromosomes. So that was a very important development. He developed a dye or a stain that binds to the chromosomes and makes them visible. And that's when we could really start looking at the cell cycle and cell division. So we're able to see the differences in the different stages of the cell cycle. And this is what it looks like. So on the top you have plant cells in the earlier stages of mitosis, and on the bottom we have plant cells in the later stages of mitosis. You can see the nice clear cell walls. It's kind of why I like plants, they're kind of easy to decipher. Nice clear cell walls. You start to see chromosomes condensing and becoming visible, and even more so, and even more so. And then here we have our chromosomes lined up at the center of the cell. You can see the cell has elongated, become longer. We start to see then the chromosomes are being separated at different ends of the cell, the bigger cell, and then we finally have division into two cells. So here's the life cycle of the cell. And just like humans, right, you don't spend your entire life reproducing, I hope, or even practicing reproducing. A large part of the human life cycle is spent on doing things besides reproduction, and it's the same thing for a cell. So if you look at this, this is the life cycle of a cell, and you can see interphase is the part of the life cycle of the cell where the cell is not going through mitosis or cytokinesis. It accounts for about 90% of the cell's life cycle.